just a couple of applications. We haven't, since our validation work is nearly complete, we haven't rolled this out for routine screening yet, but we have done a few ad hoc applications. So that's where we'll be going. As I mentioned, I'll start with the technology. There are several impedance-based instruments that are available. They all work on the same fundamental principle, which is there are thin film gold electrodes that are present in every single well, and they're interdigitated. The configuration varies widely across vendors and application, and I'm showing you one that's a fairly simple form. But essentially what you have then is these electrodes are then covered by a cell layer, and that cell layer forms a resistive barrier to current flow between the electrodes. Now, we don't call this a resistance assay because it's not a direct current that's passed between the two electrodes, but rather an alternating current, and that'll become important in a moment. So since it's alternating current, it's called impedance, not resistance. A second important sort of starting point is that the current that's applied between the two electrodes is exceedingly weak. The point is not to excite the cells. We don't have any evidence that it excites the cells. It might, but there's no evidence of that. Really, the only point is to apply a small enough current to find out what the path of the current between the cells is, or between the electrodes is. So here you see three possible paths. It can go underneath, through, or between the cells. And that would be sensitive then to cellular events like changes in morphology for movement through the cells. Cell-cell contacts would be sensitive to changes in cell contacts. And then cell adhesion would be detected by changes in this current flow underneath the cells. So it's a sum of these cellular events. All of these can be detected in an assay. Now, the relative proportion of them kind of depends on a number of things. First, what kind of assay you have. We're going to be going today towards cardiomyocyte beating, so that's going to be heavily involving morphology changes. But all of these would be in play. So it's not only the cellular assay you have, but also the instrument. And let me describe that. So there are three instruments that are available, or three manufacturers that are most well known. The first is Applied Biophysics, and their ESIS instrument is highly flexible, a lot of different applications, a lot of electrode configurations, a lot of well numbers, really highly versatile instrument. And one of the applications that I find most compelling is they are able to calculate resistance and capacitance measures and really focus in on that, and have applications for looking at tight junctions, which I find to be a particularly compelling application. Another one that I find interesting is the way that Cellular by Molecular Devices has applied this technology. They use, again, I mentioned this was alternating current, so they use 7 different frequencies of alternating current. And those currents span, those frequencies span 6 orders of magnitude, so a tremendous breadth and frequency. And all of that data is then transformed into a single trace, a single output that does something that's really stunning. It can detect GPCR signaling, whether it be through GI-coupled receptors, GQ, or GS. And that's pretty stunning in and of itself. But then it does something even more remarkable, and that is the trace that you see, that transformed trace from all of those frequencies, is processed in a way that the trace gives you a qualitative readout as to whether it is GI, GQ, or GS, which is really stunning. We've done a lot of work on that, but I won't show any of that today. Now, the latest iteration of this comes from ACEA with its xCELLigence. And here, rather than focus on the many different frequencies, as was done with the cell key, they've narrowed it down a bit to really focus on faster processing, and whereas CellKey was every 2-second updates, xCELLigence is 12 milliseconds. So you can really get to fine gradations in signaling and really resolve cardiomyocyte beating. So what I hope to accomplish with this slide was to show you that it's clearly label-free. There's no need for a dye, radioactivity, or any other label. So that's where most people stop. I wanted to also convey that it is highly flexible and we should expect to be able to do a lot of things with it. Now, most people would stop there, but I want to show you one more slide on background, and that is what I think is equally important to the label-free, and that is its high sensitivity. And that comes from some pioneering work done by Applied Biophysics, and in some of their early work, they were using small gold electrodes. They were discs embedded in the bottom of a well. And they would plate cells on one side and watch as they spread. And as you would imagine, as they spread and cover both electrodes, the impedance went up until they became confluent, and then it plateaued. They were concerned, though, when it plateaued, it was no longer a smooth trace, but became rather rugged. And they were concerned that that was a noise in the instrument or some other problem. So they characterized this and found that, in fact, that ruggedness was eliminated by depleting ATP, so it was energy dependent, or by blocking actin polymerization. So this was a specific cellular phenomenon, which they termed cellular micromotion. And as such, it became not a problem, but a tool. They could characterize their resolution of cellular micromotion and found that they were detecting changes in cell diameter on the order of 1 nanometer. To put that in perspective, a comparable assay for cellular morphology changes in real time on a live cell done by light microscopy would have resolution on the order of 250 nanometers. So this is orders of magnitude more sensitive. To put it in the perspective of a cell bilayer is about 3 nanometers. So this is done at 1 nanometer in diameter mode. So this is, as I showed in the previous slide, it's not a rounding up. That cartoon was misleading in a lot of ways. It's rather just an exquisitely subtle ruffle of the membrane is enough to be detected. So that really should change the way some people think about this, from morphology changes that are real rounding up to just really slight ruffling of membranes would be enough to be detected in some cases. So 2 orders of magnitude increase in sensitivity. What you should expect out of that is not only label-free, but a lot of new opportunities. And I mentioned one of those was GPCRs for the CellKey, but certainly this kind of sensitivity should be easy to detect beating cardiomyocytes. Should have sensitivity to spare. So to summarize the features of impedance assays, they are exquisitely sensitive. They can handle whole cell in a very robust way because of this sensitivity to spare, if you will. They're label-free, so no need for transfection or dyes or anything like that. It's real-time, so you can get continuous monitoring. And diverse applications. All of this comes together to give, in my experience, robust and versatile assays. So that's a background that I hope will help with impedance assays. Now I want to move specifically into the XCELLigence Cardio. And I want to orient you by giving you a couple of screenshots of what the data look like. This is an experiment, just a screenshot from an experiment where I was comparing rat neonatal cardiomyocytes grown in glucose versus galactose. Those are the 2 different colors. And you see that you seed the cells at time 0, and the impedance, that's the baseline for impedance. And then I let them grow, it looks like, for about 20 hours before I started a continuous read. And they sort of plateau as they adhere and become confluent. Notice the units here, cell index is going up to about 4. If the cells were to die or to detach, this would go down. So you have this continuous measure of cell health. It's not cell toxicity or viability proper because it also includes an element of attachment. But it is a very easy and useful additional measure of cell health. So that's what a user will see, general cell index or impedance along the way, and then there are beats embedded within this cell index. So now what I want to show you is a live trace, and I know that this won't come over well on a webinar, but I'll do it nonetheless. So what I'm about to show you is just 4 wells, and then I will click on this monitor button. That's what the M stands for. And if you were in my lab on any Thursday or Friday, you would see cells beating in the following way. So here are the 4 wells. I then click on monitor, and I know that the update rate is a little slow, but on my screen they are beating continuously as you go across. Now if you look up and down, I see a glitch in there because this is just a screen capture, but I also can see that the beats are not aligned, and that's because the individual wells are not synchronized. There would be no reason for them to be synchronized between wells. But within each of these 4 wells, you can see the 32,000 cells form a single beating unit. And so that synchronization and the spontaneous beating of these allow for a well-based assay. So they beat fairly consistently. They'll keep beating like that for days. And let me show you that. Oh, I just inserted this based on some questions I got on a seminar from Monday. So I'll pause here. This is an experiment that I borrowed from Lian Gao, who was at Roche when he did this experiment in Kyle Colleget's lab. And here you see xCELLigence on the left and an electrophysiological measure on the right. That's a whole cell current or an extracellular current with MEA. And what you can see is blebbistatin blocks the response in xCELLigence. It is an inhibitor of myosin 2. It does not block the action potential, and that's important. What it tells you is that the action potential is still happening here, but xCELLigence does not detect current flow related to ion channel openings and all of the features of an action potential. That can be happening, and there's no signal. But if you block the myosin, the contraction doesn't happen, and so you see very little response. So I think that really drives home the point that while we are using a very weak current passed between 2 electrodes, the current we're detecting is just looking and detecting morphology changes. So this is essentially a contraction assay. All right, now with that out of the way, I hope that really focuses on what we're detecting. We're detecting beating, if you will. Now I want to describe now a little bit of assay development work. I previously showed sort of a raw live trace. What you can get out of this is data points, and I've plotted here individual data points every 12 milliseconds connected by a line. So you can export that, and I know that Chantest is using this, and they have their own algorithms to then calculate, say, rate, amplitude, rise slope, fall slope, a number of other things. So you can do that if you like. You can build algorithms and work with raw data. Alternatively, the ACEA software includes something that allows you to extract the rate of these beats. And here what I've plotted is the beat rate over time. Now, in this experiment, I was using neonatal primary cultures, and you can see that they start to synchronize and beat as a unit at about 24 hours, and then by 48 it's pretty stable and lasts in this experiment up to 72 hours. Here's another experiment, and in this experiment it goes out to 96 hours, and in this case I've extracted amplitude, which again you can see they start to beat at about 24 hours, then stabilize, and will beat for at least a couple of days. So now this was these are all primary cultures, And in our hands, we were going through an experiment a week. And because the data was robust, we were able to really get quite a few different conditions in one experiment across a 96-well plate. So we looked at media, plate coatings, cell density, supplements, media supplements, and a lot of different things. And it took about 6 weeks, but we had no trouble getting to these kind of stable conditions that you see here. And we've published that, so if you are interested in rat, you're welcome to look at that. But overall, our experience was that the beating was stable, and I've only shown you amplitude and rate, but you can extract a number of other properties with the ACEA software. We wanted to look at the data in some specialized way, and we got xCELLigence to help us add some new features to that. So if there's something that you don't see here, our experience has been very good in getting ACEA to work with us to help us refine what we want to get out of there. So the initial assay development was fairly straightforward because the data quality was good. Now for screening, I'll show you a couple of additional experiments. First, we would like to remove serum so we don't have protein binding in our drug screening. And in this experiment, half of the plate at 48 hours is switched to a serum-free media. And you can see that the beating goes down or becomes highly erratic. And so we have not yet figured out a way to remove serum, and we've tried in a number of different conditions, but so far we are still using serum. Now, another consideration for screening would be DMSO concentration. We routinely have no DMSO in our media until drug is added. And when drug is added, here's an experiment where we look at different concentrations. For a 20-minute read, it's at about 0.7 where we see a response to DMSO. At 18 hours, you see it already a little bit by 0.1%. So we have settled on 0.1%. That's our routine anyway. There's no issue at 20 minutes. But we always include normalization to DMSO time-matched controls. I'll come back to the normalization after I show this next graph. Well, maybe actually two more graphs. I do want to share again experience on this, and so I'll highlight a feature that we see prominently in rat neonatal cardiomyocytes, and that is that rate and amplitude are in many cases change in a compensating way. And the first example I'll show you of that is cell density. So here I've plated 16 to 40,000 cells. And as you plate more cells, the amplitude goes up. And I don't know the reason for that, but I'll speculate that as you have more cells, they give you more force of beating, and so you get bigger amplitude on your beats. And I'm saying there by with that explanation that amplitude is the primary driver. I don't know that, of course, but it would make sense. Then, as you see, as you place more cells and they beat harder, they beat slower. That would fit also with the widely studied phenomenon that neonatal cardiomyocytes have a negative force-frequency relationship, such as when they beat faster, they beat with less force, and of course the reverse as well. And the explanation for that is that normally as you beat faster, you get more calcium coming in, and that then leads to more calcium in the sarcoplasmic reticulum. And under normal conditions, that additive calcium would lead to stronger contractions. But in the case of a neonatal cardiomyocyte, the sarcoplasmic reticulum is not well formed, and so you don't get that feedback enhancement. So this all fits with the biology, but it complicates the interpretation. Not in a prohibitive way, but just it's something that you need to be aware of. And if you're aware of it, I think it's straightforward to handle. Let me show you a couple more examples of this compensation, as I call it. And there are two examples built into this case. The first is changing from glucose to galactose. As you go from glucose to galactose, they seem to beat with greater amplitude. But when you have greater amplitude, you have lower beat rate. Also built into this is I've plotted this. This plate was split down the middle between glucose and galactose. And then I've plotted all the data by either edge row, one in from the edge, two, or the center. And going back up to the top, you can see that the edge rows beat with lower amplitude and higher rate. Again, just as you would expect. And that's true whether it's glucose or galactose. Now, we don't use edge wells in our experiment, but as I will now explain in the next slide, it would be fine to do so, I think. And here, let me show you why. So this is now we're going on to a little more data quality here. For drug screening, this is a set of diverse ligands, and I'm showing you only one experiment. And this is the average of 3 wells. Each well for a point comes from a replicate plate. So they're not side by side, but instead on replicate plates. And what you see here is good variance in terms of standard error. You also see good dynamic range in terms of high and low potency, good dynamic range in terms of positive and negative effects on beat rate, and good dynamic range in terms of graded positive and graded negative effects. Now that is one experiment. Replicate experiments on multiple days. You can, we've published this slide also. You can see that in the publication. I'll come back to variance in a different way in a couple more slides. I do want to pause here and again focus on our experience. Our experience is that in replicate experiments, if the beat rate here is 50, in the following experiment, if it were 60, that wouldn't affect the potency at all. And so then going back to the edge effect that I showed you, so let's see if I can go back. These data are quite tight. So if you normalize to the beat rate at time 0, if a well is beating at 40 at time 0, at 20 minutes, it will be beating at 40 again. So you can normalize in that way, and that overcomes some of this edge effect. Just to be over the top in terms of our data quality, we haven't been using the edge rows for anything other than the repeated control that we include in each experiment. So that's a good way, that we find that to be a good way to use that. Right, so then our normalization, and this came from discussions with Liang Gao, who was at Roche at the time, and we both observed, and he was using mostly the human cells, we both observed that it mattered, the beat rate was consistent from time 0 to time 20 minutes or 2 hours. So since that was so consistent, we normalized, we have adopted, like Liang had, normalized to time 0 and then normalize again to percent of DMSO time matched control. So in future slides, you won't see rate proper, you'll see percent of control. Now, so we're sort of past assay development. You'll see data quality is very good. We're into assay validation now, and for this, we wanted to validate the assay. We need compounds with in vivo data. We selected 50 compounds, 9 negative ionotropes, 9 positive ionotropes, 21 negative ionotropes, and 20 inactives. You can see by that selection criteria, we're interested in predictivity of contractility. If you are interested in arrhythmicity, Liang Gao's paper, I think it would be a great place to start. So we picked these compounds with in vivo data. Most of it's clinical, but some of it is AstraZeneca development compounds. And we used the same 50 compounds that had been used by Alex Harmer as he validated his ion optics assay. And for that, he had used canine adult cardiomyocytes. So then we repeated with rat primary cultures. I'll show you some of that data. And Xiaoyu at ACEA used the same 50 in human CDI cells. And that's a collaboration run by Clay Scott here at AstraZeneca. Clay, because he was running the collaboration, got the task of bringing all the compounds together, all 50 of them, aliquoting them, and then labeling and coding them blind. So neither Xiaoyu or I knew what they were until after we had reported our data and made the calls on whether they were positive, negative, or so on, and potencies. And I'll show you that data now. This is from the dog ion optics assay. If you're not familiar with that, the way it works is it essentially is looking at using microscopy to look at and measure the shortening of sarcomeres. Now it's a little bit different because these are adult dog cardiomyocytes, and they don't beat spontaneously. They have to be stimulated to beat. So a lot of differences here, but we find it to be a good assay, and in fact you can see that by the variance left to right for this dog sarcomere shortening PIC50. And the rat data correlate pretty well with it for the positives. Now for the humans, eye cells, they also correlate very well. And to put both of these on the same plot, a good correlation. This experiment includes quite a lot of variables. It's really remarkable that the correlation is as good as it is. We are looking here at dog versus rat and human. The dog is adult, rat neonatal, and then human is from IPS. We have optical versus impedance. We have stimulated adult versus spontaneously beating. We have primary cultures versus stem cell. A lot of different variables here, yet good correlation. Now, I won't show you the validation of predictivity and sensitivity data. That table is not quite finished, and hopefully it will begin writing that up in the next couple of weeks. So we will publish that data shortly. I do want to show you essentially from the rat perspective, which I am most familiar, 4 classes of responses among those compounds of the contractility set. And just to show you, again, the quality of the data, I'll show you duplicate experiments. In fact, it was 3 experiments, but I'll just show you 2 to keep it clean. And what I'll be showing you is a new format. Now I've switched to percent control time matched, and you'll see rate is blue. I'll explain why amplitude earned the green color, and then I plot cell index. And because it's percent of control, I can now plot all of those on a single trace. Let's see, other background. These are arbitrary units for concentration. I do to half log unit dilutions, and it's arbitrary units because that way I don't have to change the graph every time I do an experiment. I also should point out that every point on this will have one error bar that is standard error. You can't see the error bars, but they are there. And that's from 3 wells on triplicate or replicate plates. That's the controls. The controls, not surprisingly, are negative. The positive inotropes show an increase in rate with a corresponding decrease in amplitude and no effect on cell index. Now, this gets back to, I went to a lot of trouble to give you several examples. I could have given you many more of compensating effects between rate and amplitude. And here what happens is that there, again, rate's going up and amplitude goes down. They seem to compensate. Potencies are fairly similar. So you could use either. Now, in negative inotropes, it starts out similar. Rate starts to go up, amplitude starts to go down, but in this case, amplitude is not plateauing as it did for a positive inotrope. Instead, the amplitude is going to 0. So if the beating stops, then a rate may start to go up, but then it has to reverse. And that makes for very messy concentration response curves. And you really don't get good values if the beating stops. Whereas for amplitude, as it continues to go down, the beats keep getting smaller until they eventually stop, and so that gives very good curves. So all things being equal, we prefer amplitude because of this effect. But rate is still meaningful, and you want to plot it, and I'll show you the next example. Anytime you set up a rule, you want to break it, and that's exactly what this says. The other major group of negative inotropes had the following profile. Amplitudes went down until it stopped, but you can see here in replicate experiments, and again you can see, I would point out there are error bars on all of these from 3 wells. You know, we're probably going to change to, just for cost and ease, to duplicate wells instead of triplicate, because we've got overkill on this data. You can see here that rate has you see a response with rate before there's any change in amplitude. So this effect is independent of amplitude. So while they're generally compensating, that's kind of where you start, but it doesn't have to be the case. They can be independent. So now, let's see. What I want to do is show you a couple of examples of applying this in a drug discovery setting. So this is a project which I can't name. I can just say that it was a kinase inhibitor project. And as part of routine screening, the canine ion optics assay is a routine screen for us. And so the project submitted and found a couple that were positive. And here are the values for shortening and velocity as measured optically. And the rat xCELLigence assay from 2 experiments, here is the average amplitude data. And I would, mentally I would ask you to figure out what the multiple is to get from the amplitude number to the ion optics number. And in each case, it's 1 point something. Essentially, these are dead on. Now, but let me show you, and oh, I should back up. Remember that the dog ion optics assay is adult. They don't beat. They have to be stimulated. So there's no way you would ever see rate because you're pacing the cells. You have to pace them to get them to beat. In xCELLigence, it's different. And here's an example. This is one of the 2 experiments that's averaged to get this number. And you can see that amplitude is going to 0 for both. But the rate is changing independently, and that's new data. So the amplitude can line up very well with ion optics, and then the rate can give you some new information. Now let me show you another example that shows you some experience that's required in interpreting this data. This is from a project, again, that I can't name, but the project had seen that heart rate in green is going up. So this is from conscious dog data, and heart rate is going up here. Contractility is also going up, and it seems to mirror exposure to the compound. There's not a lot of information, but the project team sees rate as the primary concern. And so they asked me if I would, even though we haven't really rolled this out internally, if I would test, and they only wanted human data. So I said, all right, I'll use the CDI cells. And they want human data, and they want to see if I can replicate the rate effect. So they have a bias coming in, and that's what I have to replicate. And what I found, and I'll first interpret it through the project's perspective, and then I'll interpret it through my own. From a project's perspective, blue in my curves is rate, and it doesn't change at 20 minutes. Rate doesn't change at 4 hours. And rate actually, when you first see a change, it's going down at 20 hours. So rate going down led the project team to the following conclusion. Rate decreases, so the impedance data was dismissed. Now, I was stunned by that. Again, maybe because I hadn't explained it thoroughly. and hadn't taken them through it. But I'll give you a different interpretation. Again, starting now at 20 minutes, it looks to me like amplitude here in green is going up in a dose-dependent fashion. The data is fairly tight, and I would say that one would be I would be reluctant to ignore a direct effect of a compound on beat amplitude, a direct effect on human cardiomyocytes. At 4 hours, you still see this effect. By 20 hours, and again, there's no effect on rate, so this is pretty interesting. It looks like it's really affecting the contraction more than the rate. So again, they can be independent variables. By 20 hours, I've changed the axis here, and now you can see that amplitude is really rising. And based on the assumption here from these two graphs that amplitude might be the primary driver, when you really ramp up amplitude, I wouldn't be surprised if you saw a secondary decrease in rate in this type of assay. Again, going back to the experience I showed in the early slides, where these things most often compensate with each other. So instead, my interpretation would be to say, you know, you have a rate effect and you have a contractility effect, but I suspect that contractility is what's driving things here. If it's not the driver, it's at least maybe a primary response. Now, I can't prove that, but I can look back at the in vivo data that this project has. It has quite a bit. And whether it be guinea pig, well, in guinea pig, sure enough, contractility was more of a robust change than heart rate. And if you look at in the dog data, as you go down in dose, you lose heart rate effects, and you still maintain contractility effects in the absence of a heart rate. And some compounds only affect contractility, not rate. So this new interpretation that is driven by this data seems to fit better. And I am currently, this is, you know, sort of, again, trying to share our current experience. I think this is likely to be valuable for the project. So where would I place this based on its strengths and weaknesses in a cascade? You know, I think that we have strong capability around hERG now, right? So we have early in vitro assays that project teams can use that in vitro data. The in vitro data is now so strong, and the SAR is so strong that we now have in silico data, and we can predict hERG data before we ever make the compound. But it all comes from having a frontline screen. For things other than hERG, we don't have in vitro frontline screens like we'd like, at least not high enough throughput to be able to drive SAR. So for hERG, we've shifted from screening out hERG to designing in safety. And I think we'd like to get there, and that's why we're focused on contractility here. If we could get a frontline screen, it doesn't have to be perfect. What it does have to do is give us that paradigm shift such that we can screen more compounds and begin to design in and not replace in vivo contractility or ex vivo contractility, but enhance it and work together with the existing cascade. So where would I place this? Well, currently impedance assays are only 96 well. I think that needs to change. I'm wondering if people may go with the flipper type assays because they have 3D4. But I'm concerned about that for two reasons. And I probably wouldn't favor internally a flipper assay because I think that it's not label free, and when you start introducing a dye, particularly a dye that binds calcium, that could affect the binding. So I think there's going to be a strong place for flipper, but I think we need to appreciate that these dyes can have effects. And here I've tested a couple in the xCELLigence assay, and where you can see the effect of the dye itself, and you can see that some have it and some don't. And you could probably manage this, but I'm not sure that's where you want to be, at least for a frontline screen. Now, again, thinking about contractility and a frontline screen, what you'd like to do is have your first screen, in my opinion, be inclusive. So if you wanted to detect the action potential, you might use something that's ePhys-based. If you want to detect something that's downstream and includes action potential and calcium, you could use a calcium-based dye assay. And I think if you want to include contraction, the impedance assay would make a good frontline screen because an exchange in action potential, calcium flux, or contraction would all sum in a response that would be detected in impedance. So I see this as an opportunity to get a downstream endpoint as a frontline screen, certainly robust enough and it's label-free, and then begin to understand compounds that have effects, and then pair this data with mechanism of action studies that would allow you, if your hunch based on a positive was that it was effect on some particular property, you could go to additional assays. So I don't see the question as which is the best assay out of these, but how do we stage them in a cascade that shifts us to frontline screening and changes the paradigm for cardiotoxicity evaluation. So to summarize, rate and amplitude can be independent measures. We do see them change independently. In many cases, they're compensating, and so you need to think about which one is the driver there and include that in your interpretation. The data quality is very robust. We need to back off on our testing because we have overkill in our data quality. The validation data looks very good. Some of our misses come from the fact that we don't see beta adrenergic antagonist because there's no tone. We use dialyzed serum, and so there's no stimulation of adrenergic ligands there. So the validation looks good, even though I haven't shown all of it. From a mechanistic perspective, we think this is well placed as a frontline screen, and it will need to be paired with additional assays to look at mechanism of action. So I should acknowledge some folks. Xiaoyu did the human data in the validation set. Sarah is a postdoc and fortunate position in that all of her work will be published. She's able to collaborate, and so I hope to submit. She's been here about 6 months and hope to submit her first paper in the next few weeks. And if people have interest there, we're happy to work with folks. So with that, I think that covers what I had to say today. I'd be happy to take questions.
Okay, thanks very much, Matt. That was a very nice presentation. We do have a few questions from the audience submitted earlier through the chat. I would encourage people, if they have any additional questions, to submit them now. A couple of general questions. First, on the parameters, you focused on rate and amplitude. Are there other parameters that you see as useful or that you've seen others using? I've focused less on rhythmicity because I really would like to first get contractility. Sorry, this is not a measure of contractility. I want to be very clear, but this could be a correlate of contractility. That's where we're headed first. We have processed the data so that I can get these types of curves here for not just rate and amplitude and cell index, but I can get all of the parameters that ACEA currently gives on a single percent of control plot. But it becomes overwhelming so far, and I haven't. What I'd like to do is find some examples of compounds that change only rise slope or only fall slope. But so far, I just haven't had the time to look into that. Okay. So you actually kind of alluded to this in your answer, but I want to explore a little further. You showed a nice correlation between the amplitude and contractility. Care to speculate at all on the mechanistic implications of that? So between contractility and amplitude, is that the question? I believe so, yes. Okay.
Well, so we know that this is measuring contraction, right? So if you poison the myosin, the signal goes away, even though the action potential is still there. So it seems fairly safe to believe that this is a contraction that you are measuring. And the strength of that contraction seems to correlate with amplitude. Where you get into a bit of gray area is that the word contractility, yesterday I listened to an expert panel on contractility, and one person defined it one way, a second person defined it another way. The third person chose not to define it, but to say we need fit-for-purpose assays that will give us the answers that we need and the ability to investigate it. So it's to say that this is contractility is an overinterpretation and would be fraught with a lot of danger. So what is it? It's an assay that measures contraction of these cells, but it doesn't have all the afterload and preload of in vivo. So it's going to have to be part of a cascade. And I don't think we want perfection to be the enemy of good. We need a frontline part of our cascade. We can't have dog assays being our primary screen. It's just unsustainable drug discovery. So that's not an answer. That's kind of my take on it.
Okay, thanks very much. We had a couple of questions that were kind of related to looking at the overall cell index rather than just the beating rate. So one was very specific in your assay development section. When you looked at serum effects, the question was, did you see an effect on cell index with serum, or I guess that would apply also to DMSO, so not beating, but overall cell index. And then the related question is, have you looked at hypertrophy?
We have looked at hypertrophy, and I don't have anything exciting to offer. And as for cell index and serum, the problem as you go down in serum is that the beats are not as robust or consistent. So I was less interested in the cell index. What I really wanted was consistent, stable beating. And so that's how we drove our assay development.
Okay. Also, a very specific, in the assay development, the very first dose response slide you showed, which had the nice dynamic range and so forth, it showed the Bay-K effect. Is that in the rat primaries?
Yes.
Okay, that's rat primary data. Okay, that was the very simple question. And finally, just kind of a clarification, are then you using the data you showed, amplitude and or rate as the primary drivers for screening, or a combination of the two, or on a case-by-case basis?
We aren't there. In a big company, it takes a while to get the process and all of the. There's a lot of process involved. So no, we're not using it as a primary screen. I hope that we will. I think the data quality is fantastic, and I think that it gives a new window. But again, it doesn't answer all the questions. What it does is, I think, add to our capability. And so part of the process will be, once the validation data is complete, we'll have to have discussions about how we're going to pair it. And then a good experiment leads to more questions than answers. So we'll have to figure out, how we're going to supplement it with additional assays. What's going to be the trigger for this assay? What's going to be for a positive or negative response in various things? What is it going to trigger as a follow-up assay? So we'll get there, but we're not there yet.
Okay, thanks very much. I think we're at the end of our hour session. I'd like to thank all of the attendees, and especially you, Matt, for your very nice presentation, and remind people that you can access this presentation later if you want to share it with colleagues, and also if you have questions that weren't answered, please submit them through our website through the technical support context. So thank you very much, Matt.
Sure.